Okay, we are going to go ahead and get started here and take a look at the blood vessels in the human body. So anybody have any questions at this point before I start? Okay, and I'm going to go and open this up. And why it didn't open up. Let's try this again. Now we'll try this. Okay, now we're going to work. Okay, so we're looking at blood vessels and assessments. We're only going to do blood vessels today. We will do assessments when we come back from break. So let's move on here. Now, what you're seeing right here are two blood vessels side by side. One's an artery, one is a vein, and this is usually how our arteries and veins run in the body. They tend to run side by side in parallel structures here. Looking at this, you can tell which one is which. That's the artery. It is usually 2 or 3 times larger in size or thicker. It has a much larger smooth muscle area than the vein does. There's the artery and there's the vein. Now both of them have 3 distinct layers with a lumen in the middle. This is the lumen in here, but the tissue layers are much more pronounced in the artery. For example, there's our lumen, the opening in the middle of the artery. We have a lumen in the vein. Both of these vessels, the vein and the artery, have a very thin layer lining the inside of the lumen called the tunica. Tunic comes from the word tunic, means coat. The intimate coat, the internal coat, made up of epithelial cells that line this lumen. And both the artery and the vein are going to have this lining. This is as close to non-stick surface as we can get in here, because the surface of the vein, the surface of the artery, we don't want to have anything causing red cells and platelets, for example, to bounce up against other structures and get snagged there. So we make this about as slippery as we can. So this is the tunica intima. It's made up of epithelial cells. We would call them, they're technically called endothelial because they're internal, but it's the same type of epithelial cell. Here we have the tunica media, which is the smooth muscle layer. The smooth muscle layer is the constricting layers that surround the arteries and the veins. And you can see in the artery, it's very, very large. It's very pronounced, as opposed to the smooth muscle layer found over in the vein. And then we have what's known as the tunica adventitia. This is the connective tissue that holds the vein or artery in place to the surrounding tissues. And since our artery is large, our tunica adventitia is large. Since our vein is smaller in size, it has a smaller adventitia layer. But all 3 of these layers are going to show up in both the arteries and in the veins. Now, in our bodies, we have 3 types of blood vessels. We have We have the arteries and the smaller arterioles, which are much smaller arteries, are designed to take blood away from the heart. Whether it has oxygen in it or not, it will take blood away from the heart. The capillaries are the smallest of the vessels. They are made up of single layers of epithelial cells. So they are the smallest and the thinnest of the vessels. We also have billions of these capillaries. This is where nutrient exchange and gas exchange are going to take place. They connect the arteries to the veins, and the veins are the third category here. The veins and these smaller veins called the venules. If we have arteries and arterioles, we have to have veins and venules. Venules take blood, veins and venules bring blood back to the heart. Whether the blood is lower in oxygen or whether the blood is higher in oxygen when it's coming from the lungs. But blood is always returned to the heart through the veins and venules and always leaves the heart in the arteries. Our circulatory system is a closed system. It starts and it ends at the heart. If we pump blood out the right ventricle into the pulmonary trunk, it goes to the lungs. That blood comes back to the left atria by way of the pulmonary veins. Blood that leaves the left ventricle goes out the aorta, goes through the whole body in the systemic circulation and returns to the right side of the heart, to the right atria in the vena cavas, the superior and inferior vena cava veins. And so we have it's a closed system starting and ending in the heart. And again, now we can compare the difference in size to an artery and a vein. Here's an artery, here's a vein, and here's the capillary. The capillary is one cell layer thick. That's all it is. An epithelial cell forming the capillary. See, oxygen can't diffuse very far. It can go across 2, maybe 3 cell membranes in thickness. And so it doesn't like to go much further than that. So we keep the capillary where the actual work is being done, where the nutrients are being exchanged and wastes are being exchanged, and oxygen and carbon dioxide are exchanging between the cells and the capillaries. We keep the distance that the oxygen has to go as thin as possible, because the capillaries are only one cell layer thick. The cells are only one cell layer thick. Where we have the exchange of atmospheric air to oxygen in the blood and carbon dioxide from the blood into the atmospheric air takes place in the alveoli in the lungs, which are also structures that are one cell layer thick. A problem we encounter with the alveoli and gas exchange occurs when your patient has pneumonia. With pneumonia, we develop thick layers of mucus lining the inside of the lung, increasing the distance that oxygen has to travel. It makes it harder, it makes it much more difficult for a patient with pneumonia to get adequate perfusion of their blood. They don't get enough oxygen in their air because it's not making it across the membrane, because the membranes are covered with an increasingly thick layer of mucus. Okay, there's our capillary network in between. We have the artery to the arteriole, to the capillaries, to the little venules, to the large veins again. And we see how complicated, how complex the structures of the arteries and veins are with multiple layers. But here we see the capillary with just a basement membrane and the endothelial cells that make up the capillary itself. But this is where the actual exchange of nutrients and gases takes place in these huge capillary beds, because we have at least one capillary in contact with every cell in our bodies. In some places, up to 100 capillaries. Around the alveoli in the lungs, we may have up to 100 capillaries supplying oxygen and taking away carbon dioxide. So whether you, except for the capillaries, except for a except for the capillaries, you have three layers: the tunica intima, the tunica media, and the tunica externa. Now, the tunica media is the smooth muscle layer. It's also very elastic. It will, in arteries, it will give as blood surges through there. It is in the tunica media layers that we have sympathetic nervous system control over the diameter of the blood vessel. Vasoconstriction occurs when the smooth muscle surrounding an artery or an arteriole constricts, it squeezes. The muscle contracts, and so the opening of the artery shrinks. It gets smaller. And so we reduce the flow of blood going through there. That's called vasoconstriction. It's caused by the neurotransmitter norepinephrine. That's one of the two neurotransmitters of choice in the sympathetic nervous system, and the other one being epinephrine. Sympathetic nervous system innervates the muscles around the arteries and arterioles and can cause vasoconstriction by causing those muscles to contract. Or if we stop releasing the neurotransmitter, the vessels will dilate. Vasodilation, meaning blood vessel getting bigger. Take away the neurotransmitter, the blood vessel gets bigger. Add the neurotransmitter, the blood vessel shrinks. And so we have our our vasoconstriction and vasodilation taking place in the arteries, but primarily in the arterioles. And then, of course, the outer layer is where we have the connective tissue. Why do we have vasoconstriction and vasodilation? Because we don't have enough blood in our circulatory system to fill up every vessel all the time. We can only fill up certain vessels. So we restrict flow to other regions. And we send blood where it's needed to get rid of heat, to get rid of, to reduce heat loss, to bring nutrients, to bring oxygen, to pick up gases, CO2, for example. We send blood to where it's needed. We don't send blood to every cell all the time. Every cell gets supplied, but we can restrict or enhance the flow to different parts of our body. Now, within the arteries, we have three types. We have the conducting arteries. These are the big ones, like the aorta. Remember the aorta from looking at the heart last week? You saw that the aorta was a very thick vessel. It was usually somewhere between 1/4 of an inch to 1/2 an inch thick. It was thicker than the pulmonary trunk. Much thicker than the vena cavas, if we could have seen them. The conducting arteries, like the aorta, have the greatest amount of smooth muscle area, as well as a great deal of elastic tissue. They have a great deal of give. They have to deal with pressure changes from shooting up to 120, 130, 140 millimeters of mercury, dropping back down to 80 or 60. When we deal with our blood pressure, when we say our blood pressure is 120/80, we have that surge of blood. When the pressure, when we go up to 120, we drop back to 80. The arteries, the elastic arteries have to respond to this. The pulmonary trunk, on the other hand, only has to go from, maybe 5 millimeters to 25 millimeters. It doesn't have as near as great a range, and the pressure isn't nearly as high. But those are the conducting or elastic arteries. They're the big ones. The distributing arteries, or what we call the muscular arteries, go out to the individual organs. Like the renal artery goes to the kidneys, and the hepatic artery goes to the liver, and the splenic artery goes to the spleen, for example, or the pulmonary arteries go to the lungs. These are the ones that take blood out and distribute it to the various organs. They're smaller. They have less smooth muscle layers than the conducting arteries. And this is and in part of the distributing arteries, some of the distributing arteries are going to respond to sympathetic innervation. They're going to be able to vasodilate and vasoconstrict. I mean, they probably all do. However, there are many more of the third category. And while all the distributing arteries may be able to vasodilate and vasoconstrict under control of the sympathetic nervous system, you know, that's the fight or flight, uses epinephrine and norepinephrine. The arterioles, of which there are many, many, many more arterioles, are heavily innervated from the parasympathetic nervous system and will respond to the release of norepinephrine or stopping the release of norepinephrine. We either release it or we don't. If we release norepinephrine, if the sympathetic nervous system releases norepinephrine, the vessel constricts. If it stops releasing norepinephrine, the vessel is no longer stimulated and it will relax. And the arterioles are where we move most of our blood around is taking place in the arterioles as we dilate the arterioles or we constrict the arterioles. Again, we don't have enough blood to fill up every vessel, so we move it to where we need it. And the arterioles are doing most of that. So now each one of these arteries, these artery types, has a very important role to play. The elastic arteries deal with high pressure and low resistance. They are very elastic. They deal with the fluctuations, as I said, and they are what keeps blood flowing smoothly and continuously throughout our body. The constant surge of blood coming out of our heart is, as it moves through the conducting arteries, becomes a continuous flow for us. We don't surge and stop. We don't surge and stop. We have a continuous flow thanks to the elastic arteries. The muscular arteries are going to, as I said, we don't have as many muscular arteries as we do arterioles, so we can vasoconstrict and we can vasodilate, but we don't have enough of these arteries to have as great an impact as the arterioles because they lead directly into the capillary beds. These are the smallest of the arteries. They go straight into the capillary beds and they will determine how much blood's going to go into the capillaries. There are the arterioles. So this shows you, this is the same arteriole top and bottom. But what we're seeing here is up here at the top, this represents stimulation. These little lines represent stimulation of neurons, sympathetic stimulation of the neurons, meaning that lots of norepinephrine is being released into the smooth muscle layer right here. And so the vessel is constricting. The smooth muscle is contracting, the vessel constricts. Pressure goes up. We reduce the flow through the vessel, blood pressure goes up. Here, you'll notice that the frequency of occurrence is further apart than it was up here at the top. So that means we have vasodilation, we have less neurotransmitter released here. So less norepinephrine means more vasodilation. And so our vessel gets wider. Same vessel, but we change the diameter of it. Now let's clear this off and go to the next one. And you can see, same vessel here, what it looks like. We either constrict or contract the smooth muscle layer like we're doing here, or we let it relax. Because that's all muscles can do. All muscles can only contract, and then they have to relax. And so we're seeing a relaxation, same vessel in here. most of our changes in the size in the vessel diameter in our blood vessels are going to take place in the arterioles. And again, this is the same vessel here and here. This is vasodilated. This is vasoconstricted. Exact same vessel. Here we see running a constant maintaining tone in the muscle. So we have muscle tone maintaining. The vessel is open for a specific amount, a specific size. Here, if we increase the tone, we have more stimulus, the vessel constricts. If we decrease the stimulus, our vessel gets larger. Okay, now the smallest vessels are the capillaries. They are 1 cell layer thick. The tunica intima is the only layer they have. They are so small that only one red cell can pass through at a time. And of course, the red cells are big squishy bags of hemoglobin. They look like a red water balloon, so they can assume the shape of the capillary and squeeze their way through. But this is the advantage of this. It's very advantageous to us in that being 1 cell layer thick, they can readily absorb waste products. They can readily absorb a CO2. They can readily give off oxygen, and glucose can be released, from the plasma. So this is where we have the easy exchange of nutrients and wastes and gases in here, because the capillaries are in direct contact with almost every cell in our bodies. The only cells they don't have direct contact with are the cartilage cells. They have to get their nutrients and gases and waste exchanged through diffusion. All the other cells in our bodies have direct connections to capillaries. Now, capillaries come in 3 types. Capillaries can be continuous capillaries. They are held together with tight junctions, and they do have openings in there because they are leaky. All of our blood vessels are leaky. All the capillaries are leaky. And so there are gaps and clefts in there where plasma can leak out around the cells. That plasma becomes interstitial fluid and then is collected as lymphatic fluid before it's brought back to the plasma. Most of our capillaries are continuous capillaries. There are more of these than any other kind of capillary. Now, the second type of capillary are the fenestrated capillaries. Fenestrations look like a sieve or like a colander. They like little tiny openings. You can see here on the illustration that they have openings in here. We use these for filtering. We use these for filtering and absorption. The capillaries that line our small intestine underneath the simple columnar cells. Simple columnar cells absorb our nutrients, and the nutrients go from the simple columnar cells into our blood supply by passing through these openings, these fenestrations in the capillaries underneath the basement layer. So we absorb our nutrients there. We also have these fenestrations in the capillaries that make up our kidneys. Here, we're forcing plasma out of the blood through these fenestrated capillaries so it can be cleaned up. And so waste products can be taken out of it by the kidneys. And so here we're seeing it going in 2 different directions, yet we're going through the sieves. We see, again, the small intestines and the kidneys are the prime locations for the fenestrated capillaries. The sinusoidal capillaries have very large openings. You look over on the right-hand side at the top, you can see right here, these are the sinusoidal capillaries. They have large gaps in here. Large molecules can go through there. Large, blood can go through there. They are not. They have. They may be fenestrated. They're definitely going to have these openings here. Now, why are they there? Well, when we want to remove substances from our blood, for example, in our spleen, we have sinusoidal capillaries because we want to, when we're taking out old red cells tend to migrate to the spleen when they're worn out. We want to process these red cells. We want to collect their iron, and we want to break down the hemoglobin. And so we want to filter all this out for us. And so we have large gaps in the capillaries, so these old red cells can migrate out. We find them in our liver, where we're busy cleaning up the material, the substances that shows up in our food. So we have these large gaps here to allow us to filter and clean and go on. Okay, now let me. Continuous capillaries held together by tight junctions, a very strong barrier, yet they do have openings in there. So fluids can go out. It is a fairly uninterrupted, unbroken lining, but there are still openings. Now, in the brain, we have we have tight junctions holding the capillary, the endothelial cells together. That is what we have to make up the blood-brain barrier. So we are very restrictive in the brain as what can leak into the tissues, the neural tissues there. Fenestrated capillaries, again, we see all the pores there, the openings, so that red cells won't leave, but the plasma can come out or nutrients can come in. And of course, there's your sinusoidal capillaries. Bone marrow, liver, lymphatic tissue, particularly the spleen. We actually can allow blood cells to leave the capillaries here. And that's what we want to do when we're getting ready to recycle our red cells. Now the capillary beds are the connection between the arteriole and the venule. Hundreds of capillaries in a capillary bed. They branch out from the arteriole, and they come back together again at the venule, and they'll spread out so they can come in contact with lots and lots of cells. And we can control the flow through the capillary bed by what's known as a precapillary sphincter. And you know what a sphincter is? A sphincter is a circular band of muscle that can open or shut a vessel or another structure, like the sphincter that controls our bladder or our anus. You know, those are rings of muscle that can close and open the urinary bladder, close or open our anus, so we can defecate or so we can urinate with the urinary bladder. We bypass. We can close off the capillary flow to these capillary beds when we need to. For example, on a cold day, you would restrict the flow to much of the capillary beds of your skin so that you wouldn't be losing a lot of body heat. Because you know our blood is hot. Our blood is at 98.6 or 37 degrees Celsius. Shooting that blood into the living tissues of the skin allows that heat to escape. If we restrict the flow, we can retain more heat in our core. On a hot day, though, we do just the opposite. We would enhance the flow. We would send lots of blood to our skin and to the dermis and hypodermis to try and radiate heat away from our core. So you know, the precapillary sphincters allow us to shut down the flow through a capillary bed briefly. So we can bypass the bed completely. Everything's still going to stay alive. You know, this is not a long-term change here, but we shut everything down for a while, then open it up and shut it down and open it up. So we can bypass the bed or we can flood the bed depending on what we're trying to do. And it's all really based on the local conditions at the time. It is not a systemic you know, it's a hot day, so we're going to shut down every capillary bed on the surface. No, we're going to shut them down localized and then open them up again. Now, if we take a look at our veins, we see we only have two types of veins. We have venules and true veins. The venules are like the arterioles, they're little veins. They're very, very porous. They have lots of openings in there. Venules will lead into the veins. The veins tend to have very low blood pressure, 10 millimeters of mercury, barely enough to move through here, but it's enough to move it barely. It has 3 layers. The thickest layer, though, is the outside layer. We tend to look at veins as what we call blood reservoirs or capacitance vessels. They hold a lot of our blood. We retain a lot of our blood in the veins as a reserve for us so that we can draw on it. We have more than adequate amounts of blood to pump through our bodies at any given time, but we have our reserves in the large veins, which does create an interesting condition. If blood pressure drops between 0 and 10 in the veins, after we've come through the capillary beds, now we're in the veins, how do we get that blood back to the heart? Because if 0 to 10 is barely enough to move, how do we get it back to the heart? Well, there are 3 mechanisms that we use. We have what's known as a skeletal muscle pump, we have the respiratory pump, and our veins have a modification inside them called the venous valves. The venous valves are valves that look a lot like semilunar valves. And as we push blood through this valve, it opens up. And then as the pressure of the blood that got pushed through drops, the blood drops against the valve and the valve goes shut. And we sort of force our blood back to the heart in that manner. So let me show you what I'm talking about here. First of all, it's a skeletal muscle pump. Most of our veins have skeletal muscles surrounding them. And as the skeletal muscle contracts, it squeezes the veins and pushes blood up towards the heart. We squeeze the vein because the muscles on either side of it contract. They get larger, they squeeze the vein, they push blood up. The muscle relaxes, the blood drops back down, except we have valves in the way that prevent that from happening. The veins are we're sort of like milking by pushing and pushing and pushing and pushing. We push our blood back up to the heart. Now the veins above the heart have less work to do, so they'll have valves in there too. These valves are designed to prevent any kind of backflow. So you see what will happen is that we're squeezing this section right here, and there's blood inside that section. That forces these valves to flip open here. Now, once we do, the blood, once the blood goes up into the next section, the pressure stays high, the valve stays open. When the pressure starts to drop in here, then the valve will go shut, so it looks like those valves down there. And we keep pushing it up the vein. Now, so that's the number one mechanism that we use here. The skeletal muscle pump is probably the number one mechanism we use to get blood back to the heart. All of our large veins have valves in them to hold the blood as it gets pushed up into each region. And it looks something like this. We squeeze a section, the blood goes up, it drops down and pushes the valve shut, it stays there until it's squeezed again, it goes up to the next section, and so on. We have backflow. We have no backflow of the blood because the valves are holding the blood in place. However, gravity and pressure are not our friends. An individual who spends a great deal of time standing, for example, is going to have gravity pulling on that blood and pulling it against the valves. And eventually the valves may fail with this constant pressure of blood pushing down on the valves. If the valve fails, then the blood will drop down into the next segment. And the added weight of that blood may cause that valve to fail. And eventually your patient may develop what we call varicose veins, an enlarged vein that's very painful, and it's visible on the, protruding through underneath the surface of the skin. So varicose veins are caused by failure of these valves. Now, hemorrhoids are a type of varicose vein that occurs around the rectum. It's two causes for this. One, the individual spends a great deal of time sitting all day, and so putting constant pressure on the veins around the rectum, causing the valves in there to fail. And you get these enlarged, engorged areas around the rectum called hemorrhoids, which are supposed to be very, very painful and ugly looking. I'll show you one here in a second. Or this can occur during pregnancy. Hemorrhoids can occur during pregnancy because the pressure of the baby, you've got 30 pounds of baby and amniotic fluid pushing on the abdominal cavity, which is also creating pressure on the veins around the rectum. And again, your pregnant female can develop complications like hemorrhoids, which are they're probably more annoying than anything else, but they're still uncomfortable. So there's our venous valve. We push the blood up, the valve flips open. When the blood pressure drops a little bit, the backflow shuts the valve like that. And all of our vessels have these valves present. Here we can see, if you look very, very carefully, let me get that. Right in here, you can just see two valve flaps in jugular vein right there. So just because gravity is bringing blood back to the heart for us, we don't want to bring too much back too fast. So we have to still maintain and still be careful of the amount of blood going through there. And so we have our valve flaps, our valves either way. Now, valves were discovered 300 or 400 years ago by William Harvey, the same individual that discovered oxygen. He put a tourniquet around his arm, very much like we would have if we're having blood drawn or if we're giving blood. When we put a tourniquet around our arm and we partially occlude our veins, the veins pop up. So once he identified the veins, he was able to take his finger and run it along the vein and push the blood out of the vein towards the heart. So he would find the vein. It would pop up thanks to the tourniquet. He'd then take his finger where the vein popped up and push the blood in that vein towards the heart. And he discovered that there was no backflow. When he pushed it out of the region, out of the vein, he discovered that there was a valve there to keep blood from flowing back into the vein. So that's how he discovered that we had valves in there. One-way valves. Blood can go up through the valve, but it won't come back. And so this was discovered probably 300 years ago and then confirmed through cadaver dissection. So, now, varicose veins look like this. We have the veins become distorted and are near the surface. They're very painful and uncomfortable for your patients because the valves have failed and blood is piling up around the valve that's still functioning in here. But the problem is, as more and more blood accumulates, it will overwhelm the valve until we keep getting larger and larger, what we call the varicose vein here. and usually they need to be cleaned out. They can be fixed, cleaned surgically. Your patients that have occupations where they are spending a great deal of time standing or sitting can develop varicose veins, or they can develop hemorrhoids. Hemorrhoids are veins that become enlarged around the rectum or around the anus simply because of constant pressure, restricting the flow of the blood in the vein. And they end up looking something like this. This is the swollen structure is the swollen hemorrhoid right there, engorged with blood, swollen tissue, very, very painful. And it usually needs, it can be treated topically, but if it gets out of hand like this apparently has, that has to be treated surgically to have it removed. So, now, okay. Now we can take a look at the models now to look at the various blood vessels in the body. First thing you see here, we're looking at the torso of the individual. This is the torso model. We're looking right at the neck, and we can see two very large veins and arteries here. Now, the first thing we're going to see is one that we hadn't thought of. This is called the left brachiocephalic vein. And the left brachiocephalic vein is very small. It only goes from here to here. That's the left brachiocephalic vein. And if you have a left, you're going to have a right. And this is the right brachiocephalic vein over here, left and right. Now, adjacent to the brachiocephalic veins on either side are the left and right subclavian veins. The subclavian vein runs directly underneath the clavicle on both sides. The left subclavian and the left brachiocephalic come together and help form and form the superior vena cava, the large vessel vein that drains into the right atria. Here we see the left jugular vein. The left jugular and the left subclavian come together to form the left brachiocephalic. And if you have a left jugular, you're going to have a right jugular. If you have a left subclavian, you're going to have a right subclavian, and a left brachiocephalic gives you a right brachiocephalic. Everything on the left side of the body is mimicked by everything everything on the left is mimicked by what's on the right. Now, adjacent to the jugular vein is the left common carotid artery. In most places in our bodies, we're going to see the artery and the vein have a common name, like left iliac artery, left iliac vein, for example, or left brachial artery, left brachial vein. But the jugular and the common carotid are one of the exceptions here. The jugular, maybe they were discovered differently. At different times, I don't know, but I know that the large vein draining the head is called the jugular vein, and the large artery that supplies blood to the head is the carotid artery. Now, another thing about terminology, anytime you see the word common in here, when you see common, like left common carotid right there, or you'll see the right common carotid over here, what that means is an artery that is, or a vessel that is common, is going to it's going to split at some point. It's going to split like this, where you have the common vessel here, and then it may split into internal and external. So when it says common, so in this case, the left common carotid artery will split at the carotid sinus into the left to the internal and external left carotid arteries. You can feel the carotid sinus on your neck if you come down in a straight line with the tip of your ear. and with your earlobes, come down to the tip of the earlobe, straight down underneath your mandible. You should feel your carotid pulse. Don't put a lot of pressure on that, but you should feel your carotid pulse there. And that is where that carotid pulse is what we call the carotid sinus. That's where the carotid artery on each side splits and goes internal and external. So common always indicates some sort of division occurring. Left common carotid branches into the left internal and left external carotid. Same thing will apply on the right-hand side. Okay, now over here is the right brachiocephalic vein, and right above that is the right subclavian vein. Again, that runs directly underneath the clavicle. Here we see the right common carotid artery corresponding, it is the counterpart to the left common carotid. What you can't see here, because this muscle's in the way, is that our left, the right jugular vein is masked by this muscle on the model. But right here, at this little tip right underneath the arrow, this blue part going up and then disappearing, that is the right jugular vein. You can't see much of it there, but it really is right there. So now, let's take a look at the overall torso model of this, the flat model here, and we can see some of these vessels in greater detail. So first of all, there's the pulmonary trunk coming up out of the right ventricle. There's the pulmonary semilunar valve right there. There is the ascending aorta coming up out of the left ventricle through the aortic semilunar valve, which is tucked away right up here, just to the left side of the mitral valve. This is called the aortic arch, where the aorta curves and then dives down to end up going down through the diaphragm and becoming the abdominal aorta. In fact, you can see, oops. If you look here, you can see where the aortic arch curves right there and continues on down, goes through the diaphragm, and now we have it continuing as the abdominal aorta here. So the aorta, of course, is the largest, most powerful blood vessel in our body. But there's the aortic arch. There are 3 distributing arteries coming off of that. The first one is the brachiocephalic artery. The next one is the left common carotid. And the third one is the left subclavian artery. So coming off the heart, the 3 openings on the heart, the 3 openings on the aortic arch. The 3 openings in the aortic arch. Here's the aortic arch right here. Opening number 1 is the brachiocephalic. That's right there. The next one is the left common carotid. And the third one is the left subclavian, right there. Left subclavian is this one, brachiocephalic is this one, and left common carotid is in the middle. Okay. Now, there's the brachiocephalic artery. It is probably the shortest artery in our bodies because it will give rise to the right subclavian and the right common carotid. there's the right common carotid. Get this out of the way here. Oh, let me get back. What did I do? Okay. There we go. Now, the right common carotid is right here. But the right common carotid is coming off the brachiocephalic artery. And you're going to see here in a second that the right subclavian also comes off the brachiocephalic. So the brachiocephalic artery is only about an inch long at best. You can see that this area right in here is where the brachiocephalic artery is located. So this is all we have. This area here is the brachiocephalic, because as soon as we get past the subclavian, we're in the vena. We are going to become the right common carotid. Right common, there's the brachiocephalic up to about here, where the arrow is, and then we have right common carotid going up and right subclavian going off to the side. So it doesn't give us. It's not a very large. It's not a very large artery at all. Okay, what else do we have here? Vertebral artery. The left and right vertebral artery is going to supply the vertebrae coming off of the subclavians. These arteries are great arteries to use when doing bypass surgery. Now, many people still have bypass surgery that use the large subclavian vein out of the leg, and it's a great choice. It has all three layers. It's a very good procedure. The advantage of using the vertebral artery is that we reduce the number of incisions you have to make. Because with the subclavian artery, I mean, if we're taking out the great saphenous vein, you got to cut that off at both ends and tie it off. And then you have to make an incision above and below the blockage in the heart. And then you have to tie in the saphenous vein at that incision above and below. Using the vertebral artery, you make one incision to cut the artery off its connection to the vertebrae. And since there are three arteries altogether on each side, you can afford one artery to be pulled away. And then you only have to tie that in to the artery in the heart below the blockage. So you're essentially making two incisions instead of four. And you're dealing with an artery to an artery, as opposed as putting a vein in place of an artery. It turns out that veins are more susceptible to plaque accumulation than arteries are. And so an individual who has bypass surgery, if they don't change their lifestyle, then they will they will end up blocking that replacement artery or replacement vein in time. So that's never a good thing. Anyway, there's the vertebral artery on the left side, and on the right side, there is our right axillary artery. This comes down off of the subclavian into our armpit. The axillar region is where we have the axillary artery. The axillary artery then becomes the brachial artery. Brachial artery is the one that we often notice, that we always use when we're taking blood pressure. We put the blood pressure cuff around our forearm, right above the elbow, and we are putting pressure on the brachial artery. The brachial artery is right there. Now, the brachial artery will then divide into two other arteries, the radial artery and the ulnar artery. Now, you can find those very easily. If you look at your hand and you find your thumb, and most of us have a thumb, you find your thumb, you know that your radius, the bone in your arm, the radius lines up directly behind your thumb. In fact, the styloid, when your hand is upright and your thumb's pointed straight up, your styloid process on the radius is pointed right at the thumb. So now you know where your radius is located. Your radial artery is on top of the radius. Your radial vein is underneath the radius. Now, knowing where your thumb is, now you just, all of a sudden, now you know where an artery and a vein is located. You also know that the ulna is underneath the radius. And that means that the ulnar artery runs on top of the ulna and the ulnar vein runs underneath the ulna. So just by knowing where your thumb is, you now know where 4 vessels are located. The ulnar artery, the ulnar vein, the radial artery, the radial vein. And so just by spotting your thumb. You also know where to take your pulse, because right behind the thumb, at the base of the thumb, is where you press against the radial artery to detect the radial pulse. Radial artery and ulnar artery right there. They split off of the brachial artery. And there's our diaphragm. So just for reference. And the abdominal aorta, I showed you that. That is a continuation of the aorta. It comes up out of the left ventricle, curves around in the aortic arch, gives rise to those 3 distributing arteries, then turns and dives down through the thoracic region and into the abdomen. And we have the abdominal aorta that runs down through most of our abdomen until we get down to about the level of the belly button. Okay. Now, in addition to the radial artery and the ulnar artery and the radial vein and the ulnar vein, we have 2 other sets of veins and arteries in our hands. So now if we look at the end of our hand, obviously there's our thumb. There's the radial artery on top of the radius. There's the radial vein on top of that. There's the ulnar artery and the ulnar vein underneath it. Now, the ulnar arteries, the radial and ulnar arteries will give rise to an arch of blood flow. And so will the veins. We have the superficial palmar venous arch. That is the blood flow going back to the heart, leading to the ulnar vein and the radial vein. And directly in front of that, we have the superficial palmar arterial arch. So they connect to each other right there. So now you know 6 vessels: radial artery, radial vein, ulnar artery, ulnar vein, palmar venous arch, palmar arterial arch. But we're not done yet. We also have the digital arteries and the digital veins in the fingers. So now you know 8 vessels: radial artery, radial vein, ulnar artery, ulnar vein, palmar venous arch, palmar arterial arch, and the digital arteries and the digital veins. There's 8 vessels that you now know just by knowing where your thumb is. In addition to being able to take your pulse at the radial artery right behind at the base of the thumb, now we know where the radial pulse is taken. So our thumb is an amazing structure here for us. Now, taking a closer look at these arteries, if we look at the arm model, for example, you can see this is the axillary artery here. It's coming off the subclavian. There's the axillary artery coming off the subclavian. The subclavian artery would be up in here. The axillary artery gives rise to the brachial artery, usually underneath the biceps. And then that will divide into the radial artery and the ulnar artery. And the radial artery here behind the thumb, the ulnar artery lined up behind the little finger. Radial artery there, There's the palmar arch right there. And there are the digital arteries there. Now we have the corresponding veins that go with it. And if we go back here, the radial artery and the ulnar artery came off the brachial, which comes off the axillary, which comes off the subclavian. So now you know 11 vessels. Actually more than that, because you're going to have a brachial vein and an axillary vein. So you're going to know 13 vessels just by knowing where the thumb is. As we start with the thumb, we get the palmar arches, we get the radial arteries and veins, we got the digital arteries and veins, we got the ulnar arteries and veins, we got the brachial artery and vein, and the axillary artery and vein, just by knowing where the thumb is. So all that's just, there it is. So anyway, and again, there's our radial artery, radial vein. The only reason we don't, radial artery, ulnar artery, and the palmar arch. The only reason we don't have the veins listed is because they don't show up on this model. So anyway, and there's the digital arteries right there. Now, if we move a little further down, we can discover some other structures here that I want to point out to you. There's the aortic arch, but notice here, these three distributing arteries coming off of the aortic arch are now visible. We've made that brachiocephalic vein transparent. So the first one is the brachiocephalic artery. The second one is the left common carotid. The third one is, the left subclavian. Now, there's the left common carotid coming off the number 2 spot here, and the right common carotid branches off of the brachiocephalic artery, as does the right subclavian artery. So we still have 3 big distributing arteries coming off the aortic arch, but we also, the brachiocephalic artery gives rise to the right subclavian and the right common carotid. We get the left subclavian and the left common carotid coming directly off of the aortic arch. Now, if we look at our heart model here, we can see the left atrium, the left ventricle. We have the mitral valve and the chordae tendineae clearly visible here. And we see there's the chordae tendineae, and there's the papillary muscles. And we found last week that the papillary muscles in the cow heart look very much like that illustration does there. The shape of the papillary muscles are very large and bulky, just like they are on the model here. Okay. And there is our interventricular septum, which separates the wall of the left ventricle and the right ventricle. We saw last week in the heart that it really was that thick. It really is that thick. We also saw that the right ventricle doesn't extend all the way to the apex, but the left ventricle does. Here we have the right atria with the right ventricle, the tricuspid valve with its 3 flaps. There's the pulmonary semilunar valve right there. And the pulmonary trunk with the pulmonary semilunar right in front of it. Left pulmonary artery, the right pulmonary artery. They are marked, indicated blue on the model because they have less oxygen in them than in the arteries, other arteries. They're probably running 25% saturation, maybe 80% saturation, as opposed to 98, 99% saturation in the other arteries. Now, there's the aortic arch again with the ascending part with all 3 distributing arteries coming off, brachiocephalic, left common carotid, and left subclavian. There's the arch. brachiocephalic, left common carotid, left subclavian. Up here, there's the superior vena cava, and that makes this the inferior vena cava down here. Both are dumping into the right atria. Here we see the pulmonary veins. The pulmonary veins are bright red because they're bringing blood back to the left atria that are fully engorged with oxygen, 99%, 100% saturation. You have pulmonary veins on the right side and the left side coming into the left atria. In fact, if you look carefully, you can see right here and here are openings for the pulmonary veins. Those are going to be the left pulmonary veins. In fact, there they are. There's one, and the other one's right there. The right pulmonary veins run all the way across the back, and they open out somewhere in this area right there. Now, let's clear everything away again. There's our superior vena cava, left brachiocephalic vein, right brachiocephalic vein, left subclavian, right subclavian, left internal jugular, right internal jugular, right axillary vein, right brachial veins. Like I said, the arteries and veins run pretty much side by side. So knowing your radial artery gives you the radial vein. Knowing the radial artery gives you the ulnar artery, which gives you the ulnar vein. You have the palmar arches and the digital veins and digital arteries. You have the axillary artery and axillary vein leading into the brachial. The axillary artery connects to the brachial artery, which connects to the ulnar and radial arteries. The axillary vein is drained by the brachial artery, which is being drained into by the ulnar and radial veins. So remember, they're going in opposite directions. So you know 14 different vessels just by knowing where the thumb is. Okay. Cephalic vein runs near the surface of the arm, and the basilic vein runs near the underside of the arm. And then we have this vein that runs around the elbow called the median cubital vein. And the arteries that would match these are not shown here. So, okay, diaphragm. This is actually, I love this. It's spelled diaphragm. We left it, apparently they left a couple A's out of the word diaphragm. So anyway, there's the inferior vena cava. The inferior vena cava is the largest vein in the body. The only reason it's called inferior is because it's underneath the superior vena cava. It's much longer and larger than the superior vena cava. This is the superior vena cava here. The inferior vena cava runs all the way down to the inguinal region. So now we have a vein coming back from the stomach area called the gastric vein. We have a vein returning from the spleen called the splenic vein. We have veins returning from the small intestine called the superior mesenteric vein. Now here's where we have a real exception in the return flow to the heart. We have a vein here called the hepatic portal vein. Hepatic portal vein allows us to bypass the vena cava. Blood coming from the small intestine contains all the nutrients that we've absorbed, with all the chemicals and preservatives and additives in there, plus anything we've taken, medications or things we've ingested that we don't necessarily need to hit the rest of our body. So the hepatic portal vein takes that blood supply from the mesenteric vein and runs it into the liver for cleaning up. We detoxify it. once it's cleaned up, once the material that's been absorbed is cleaned up, it is then discharged back into the vena cava through the hepatic vein itself. And that's what's happening here. That's the hepatic veins. So we actually bypass what you're seeing. What we do here is, where's my color? We're going to bypass the inferior vena cava. Instead of going directly into the inferior vena cava, we use the hepatic portal vein to go into the liver, and then we go into the inferior vena cava after it's been cleaned up in here, and then we go on our way to the heart. So because we just don't want to send nutrients with additives in there directly out into the blood supply to the tissues until we've cleaned it up. So, and that's what the liver does. It cleans things up for us. And there is the renal vein matched by a renal artery. Okay, now close up of this. We have the common hepatic artery, blood supply there. The celiac trunk sending blood out to the stomach. There's the gastric artery going to the stomach. There's the splenic artery going to the spleen. There is the superior mesenteric artery going out to the small intestine. And the hepatic artery here is supplying blood to the mesenteric artery. That blood will go into the capillary beds of the small intestine and then absorb the nutrients, which will then come back through this vessel right here. Will come back through this vessel here, which is the superior mesenteric vein, and then go through the portal system. But there is a superior mesenteric artery going to the small intestine. And we also have an inferior mesenteric artery going to the large intestine. And there's our renal artery supplying blood to the kidneys, and the vein here next to, adjacent to it, draining blood back to the vena cava. The gonadal arteries are sending blood to the ovaries or testes, and there are veins bringing that back. So, of course, there's the splenic vein coming back from the spleen. Now, close up on our kidneys, we can see the renal arteries in blue, renal arteries in red, and the renal veins in blue. There's the abdominal aorta with the inferior vena cava right next to it. Don't worry about these little vessels cut off because you can't tell which ones they are, because all we have left are the stubs of the arteries and veins. So we just ignore that. Besides, we've seen these in the larger illustration anyway. So now, abdominal aorta, inferior vena cava, renal artery, renal vein. Renal artery comes off the abdominal aorta. The renal vein goes into the inferior vena cava. Same on the other side. Now, the renal artery, the abdominal aorta divides just below the belly button region. And it divides into the right and left common iliac arteries. So this is the right common, here comes the abdominal aorta, and it splits right about here to the right common iliac. That's what this is here, and to the left common iliac. This is the left common iliac artery here. Now, if you know that if it's a common, it's going to divide into an internal and an external artery. The same thing applies to the vein. The artery is called the common iliac artery. The vein will be called the common iliac vein. So let's clear this and see what happens. There is the common iliac vein on the right side, right common iliac artery, the right common iliac vein. The right common iliac artery will split into an internal and external. In the case of the vein, an internal and an external come together to give you the common. So let me go back. So we have right common iliac artery, right common iliac vein, left common iliac artery, left common iliac vein. Let's go take a look at this. Now we can see where everything is located. There is our right and left common iliac veins in blue. We have the right and left common iliacs come together to give you the vena cava, the inferior vena cava. Remember that our blood flow is going this way. So the vena cava is formed from the right and left common iliacs at this point. Now the common iliacs are formed from the junction of the external and internal vena cavas on each side. So right here we see the right and left external iliacs. The right and left external iliacs come together with the right and left internal iliacs there. And let me go back, sorry. So here we have the right and left, come on, get out of the way. Here we have the right and left external iliacs here and here, the right and left internal iliacs here and here. Now they will come together this one comes together with this, and this one comes together with this to give us the common. From here up, they're the common iliac veins because they're going that direction. They're going up towards the heart. So the right and left external iliacs here and here combine with the internal iliacs here and here, and now we're at the commons on both sides. Now let's take a look at the arteries. There are the right and left common iliac arteries. There's the vena cava. Of course, this is the abdominal aorta in red. Now here we have the right and left external iliac arteries, and they've branched back up here to give you the right and left internal iliacs too. So the abdominal aorta comes down, divides into the right and left common iliacs, and then the common iliacs divide into the right and left internal and externals. So that's how, and the veins are doing just the opposite, going back up. Again, whenever you have a common, you're going to have it branching out one way or another. So, okay, now we get into the lower portion of the body. We can see the abdominal aorta right there. Here's where it's dividing right at the common iliac artery. There's the common iliac artery on the right and left side. And we can see the vena cava formed by the right and left common iliac veins. Now we can see the deep femoral arteries and the deep femoral veins. They're running right below the head of the femur. They run right below the acetabulum. This vein that runs down this way in red, I'm sorry, this artery in red is the femoral artery on both sides. That's the femoral artery running down here. We have a circumflex artery and vein which runs around the neck of the femur. This is why when an individual breaks their hip, there's often a lot of bleeding and inflammation and swelling and pain, because if the neck of the femur, because when someone fractures their hip, it's nearly always going to be breaking the neck of the femur. And when it breaks, it's very likely going to tear into the artery or the vein surrounding it. The circumflex artery and the circumflex vein run right around the neck of the femur, because there's the artery right next to it. Yeah. Here is the true femoral artery, this large one here that we see coming down our leg, and right next to it we have the femoral vein on both sides. You know, the deep femoral artery is this one that runs over here. So, in fact, there it is. You know, it is not just the femoral artery. And we go a little further down in the below the hip, and we're into the area of the great saphenous vein. It runs along the medial side of each leg. It's a very long vein, and we can do without it because we found that we can remove that vein and use it for bypass surgery as a graft. So now, again, here's our femoral artery and femoral vein in our our thigh. It runs adjacent to the vastus medialis, one of the four quads on the medial. You know, it's on the medial side of the leg. You know, or the big toe side of the leg. Femoral artery, femoral vein. If we move further down the knee to the knee, we're going to see there's the great saphenous vein. The deep femoral artery is on the lateral side, and the femoral artery is on the medial side. Now we have arteries that run down along the tibia. We have the anterior tibial artery. If we have an anterior anything, we got to have a posterior, and we do. If we have an anterior tibial artery, we have a posterior tibial artery along the back side of the leg, and we're going to have an anterior and a posterior tibial vein too. So here's our leg muscle in the thigh area, in the tibia area. There's the tibialis anterior, and this is the anterior crest of the tibia, our shin bone. Remember that sharp edge of the tibia? And running adjacent to that is the anterior tibial artery. And the posterior tibial artery is on the back side of the leg. Here we can see, here is the posterior tibial artery right here in red. There's the popliteal artery giving rise to the the posterior tibial artery. This is the popliteal vein, and the popliteal vein and the posterior tibial vein are common sites for deep vein thrombosis, because when the leg is bent at the knee for long periods of time, it's not unusual for a clot to form behind the knee and block the return flow in the vein. Usually the popliteal vein or the posterior tibial vein. And this, of course, right next to it is the posterior tibial artery. So we have the popliteal artery, the popliteal vein. We have the posterior tibial vein, and the posterior tibial artery. And as I've always said, if we have a posterior, we got to have an anterior. We had the anterior tibial vein, tibial artery on the previous slide. Right there. Now let's get back to the next slide. Here we are looking at the end of the tibia and the foot. There is our anterior tibial artery extending on down into the foot. There is our anterior tibial vein. It's on the medial side of the leg because it lines up with the big toe. There is our fibular vein. It's nice that the fibula gets something. And we have our dorsal pedis vein, dorsal pedis artery, arcuate artery, arcuate vein, dorsal venous arch, and the metatarsal arteries, metatarsal veins. And that is where we stop. That is all the vessels of the human body. Now, there should be a list. There should be a list of all these vessels in your content section of eLearn. There's also a list in the AP2 Lab Guide you can find in the content section of eLearn. So in the table of contents section, so you have all the arteries, all the veins in there. There's only about 60 vessels. And remember that everything on the left is matched by everything on the right. There are very, very few exceptions to it that you encounter here. Anybody have any questions on anything? Okay, then I'm going to get us out of here. We will not be having any tests the week we come back. I can tell you that from break. And we will move on. So we're done with this next week. In 2 weeks, we will do the assessments and we'll be done with the second unit in lab. So the assessments, how to take blood pressure and pulse and stuff like that. So, okay, I'm going to get us out of here then. And you all have a very nice break.